when we think plant protection, we often think, stopping things from coming in and hitting the plants. We don't often look at it the other way around, which is the way I like to attack it. How do we promote the plant health? Where do we start? We start from the soil, we start from our varieties, the way we grow these things. Another challenge we have, and everyone's facing this right around the globe, is the evolution of resistance to our chemistries, whether it's an insecticide or a herbicide or a fungicide.
Welcome to the Crop Science Podcast Show, brought to you by Wise Genetics. I am Brian Ernell, and I'm coming to you from the campus of Oklahoma State University. Today, I'm excited to have on Dr. Anthony Young. Dr. Young is a senior lecturer in crop protection at the University of Queensland's Gatton campus. Is it Gatton, or how do you say that right?
We say Gatton, but it doesn't really matter.
Gatton, that's even better. Gatton campus. His research is focused on the characterizing and managing plant health problems. He's used molecular biology to work on a wide range of crops and a wider array of their pests and pathogens. And first, Dr. Young, thanks for joining us. I know it's early where you're at, and it's hot where I'm at.
No, thank you very much for having me. You've caught us in our middle of our winter. It's 6 a.m. out here, it's still dark, and it's a pleasant time of the year in Australia.
I can imagine. So, as I always do with all my guests, I ask for you to share briefly about your path to your current position. What brought you into, why do you dive into a career of crop protection?
It's a good question, Brian. I don't know if anyone actually dives in. I think most of the plant pathologists are accidental plant pathologists. I grew up on a cane farm in northern New South Wales. It's a small fraction of the Australian sugar industry. I was the sixth of 7 children. I was the first of those children to go and get a university education, only because I didn't know what to do. I did a biology degree at Macquarie Uni in Sydney. And after my honours year, I was picking cotton out at Moree, driving tractors, fixing up machinery, farm work. And my old supervisor told me that there was a PhD scholarship in a sugarcane disease, Ratoon Stunting disease.
Mm-hmm.
And for me, that meant 3 years of, I was getting $800 a fortnight, tax-free, sign me up. It's funny because I never selected plant pathology. I was always more into zoology, entomology. But in saying that, if I could go back and talk to that younger version of Anthony, I'd say get into plant pathology because it's a magnificent field. That's how I landed in plant pathology.
That's fantastic. I love, I'd say a lot of us in academia are about the same. Part of the reasons I got a master's and a PhD is because, I couldn't get a real job, is what I tell some folks. Nobody wanted to hire a kid to be an agronomist who hadn't lived on a farm. So, the academia worked its way in. That's great, Anthony. So, talk about some of the work that you're currently involved in. What are you doing down there?
So I've got a job at the University of Queensland where I teach plant protection. And so a lot of our students graduate to become agronomists. I guess my role is to make sure they have some concept of the things that are going to hit the plants that their clients are going to employ them to look at. Some of the challenges we have is the changing climate. We're seeing different pests and diseases in different areas that we haven't seen them before. We're seeing different impacts on those because of the stress associated with the plants. So part of my role, research-wise, is to try and disentangle some of these relationships, to try and work out what's going on and how best we can mitigate it. And when we think plant protection, we often think, stopping things from coming in and hitting the plants. We don't often look at it the other way around, which is the way I like to attack it. How do we promote the plant health? Where do we start? We start from the soil, we start from our varieties, the way we grow these things. Another challenge we have, and everyone's facing this right around the globe, is the evolution of resistance to our chemistries, whether it's an insecticide or a herbicide or a fungicide. And we've fallen into the trap, over the past sort of, let's say, 60, 70 years of just you have a problem, you spray something on it. But
Yeah.
that's not going to work. That's going to pull us apart eventually in our whole production systems if we rely on these simple modes of action. So yeah, I guess my research is looking at those relationships, those biological relationships with whether it's a virus or a bacterium or a fungus and the host, and to work out why that relationship breaks down.
That's fascinating. So I want to kind of go back a little bit more on a generic conversation. Is that I'm going to assume a great bit of our listener group's not familiar with cane. And if they are, they're going to be familiar with either Brazil cane, Louisiana cane, that Southeast U.S. cane. And both of those are very different. What does the Australian cane production system look like?
Okay, so it's almost 100% mechanized. I grew up on a cane farm, as I mentioned. I spent my 14th birthday manually planting sugarcane. Got very fond memories of that, working hard while my mates are
Yeah.
down the beach having fun during our springtime.
Yeah.
So we're a small but very efficient industry. So I think Australia might be the third highest exporter of raw sugar. That's behind Brazil and I think Thailand. Um, but yeah, we moved from hand cutting back in 1974. My father was one of the first hand cutters of sugarcane. So he left school at
Oh.
14 to cut sugarcane by hand. But in the 70s, we moved away from the hand cutting, and we actually pioneered the original sugarcane harvesters, the mechanical harvesters, which have
Hmm.
been adopted worldwide. Our industry is relatively clean, so we're reducing the amount of burning that we're doing in the industry, and a lot of that is finding new markets for the thrash. When we say the thrash,
Hmm.
if your listeners think about sugarcane, it's a giant grass. It has dead leaves on it.
Hmm.
Typically, when we go to harvest that cane, those leaves, that thrash, can interfere with that harvesting process, and so historically we've just burnt the cane. But now what we're doing is we're trying to reserve that thrash, and we bale it up and we
Hmm.
sell it into different markets as garden mulch, etc. Compared to a lot of other sugar industries, we have fairly high CCS. So if you go to, for example, Indonesia, their CCS, and when I say CCS, I mean commercial cane sugar, and that's essentially the proportion of the plant that's sugar that you can recover.
Hmm.
They might be sitting at about 9%, whereas ours is sitting at about 13, 14%. So we have high sugar, and like I say, a highly mechanized industry from our transport, our harvesting, our crushing of the sugarcane, all mechanized.
So I'm kind of intrigued the removal or moving away from the burn because for me I think about burning and that also handles part of the disease triangle by removing lights amount of residue that could easily have spores and others. How has that moving away from burning more of a baling? Because I would, if I imagine the baling, there'd be more residue left or more potential small parts of residue left. How's that impacted or has it impacted disease management within cane production?
Yeah, so some of the older growers get really concerned about what they call army worms, and they're not quite the fall army worm, but they're let's just call them army worms, right? They
Okay.
can live in that residue that you're talking about, and they can come and eat the green shoots as the. Oh, so your listeners know when we harvest the sugar cane, when we cut it, it's like mowing your lawn back home.
Mhm.
You don't replant your lawn; the cane grows back up again, and these are what we call ratoon crops or resprouting crops, right? So they get concerned that these army worms are going to eat those ratoons, sort of thing, and so many a farmer I've seen burn the thrash that's there, completely wipe out the young ratoons, burn off those new leaves. I said, well, tell me if the army worms are doing less damage or more damage than what you've just done.
Yeah.
Um, the main sugarcane diseases probably wouldn't have the retention of the residue probably wouldn't have a big impact on those guys. So
Mhm.
the if we look in the Australian system, well, the number one disease, and I'm sure this is the case worldwide, is Ratoon Stunting disease. It's a bacterial disease. We'll talk about this a bit later on. It lives in the xylem vessels. So it's not impacted by the burning. The other diseases, we get sugarcane smut, which forms a smut whip. Again, those spores, there are trillions in the air. The burning doesn't really have any impact on that. There really are two major ones. The last one is chlorotic streak disease, which is a waterborne disease, which I've done a fair bit of work on as well. Again, that's not impacted by the burning. We have a lot of minor leaf spot diseases. We have rusts, which can be bad in some circumstances. But the retention of that trash actually makes much more biological sense in terms of
Mm-hmm.
improving that soil health. And I didn't talk about some of these soil-borne diseases, things like pachymetra or the nematode impacts.
Mm-hmm.
And as your listeners would know, the more organics we get into that soil, the more stable and more antagonistic a microbial community we build, better soil health, more food for earthworms, plus, 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 plus. So, yeah, it's much. There's many more benefits in retaining the trash than burning it.
Oh, absolutely. So Dr. Young, I'm a soil scientist by training. I work in soil fertility, and every trip to a cane field, especially post harvest, hurts my heart a little bit. As just far as the mechanical nature of most cane production and how it does impact the soil. I mean, there's a lot of weight, there's a lot of movement, and there's a fair amount of destruction when you start talking about soil structure, and then you're burning and removing, so there is no carbon going back to maintain any of that structure, especially over time.
Yeah.
How long is a Queensland or Australian cane crop in field before that cane crops, I guess not out? How many years do you get out of a single planting before you rotate, and what's your rotations?
Okay, so the first question touches on our topic, Ratoon stunting disease, I suppose, in some respects.
Hmm.
Almost all of Australia, like all of Queensland, they grow their cane in a 1 year cycle. So they grow the
Hmm.
sugarcane up for one
Okay.
year, then they harvest it, and then they grow it for another year, then they harvest it. In northern New South Wales, 80% of our crop is 2 year old crop. We grow it for 2 years and then harvest. And that's because we're further south, which for an American audience would be further north. It's cooler, less sunshine, etc. Um,
Hmm.
Less heat, I should say. So in Queensland, it's a 1 year old crop, and most farmers will aim to get a plant crop, first ratoon, second ratoon, third ratoon, if they're lucky. So let's say four crops, so 4 years or 5 years. But then what happens is the yields start deteriorating. So typically your plant crop and your first return are pretty
Hmm.
good, but then those yields start going down, and then the farmer's looking at the cane, it's a bit scratchy and they're not sure and they don't think it's profitable. So they will hook it out, they'll plow it out, and they've got two options then. They can either replant that year with sugarcane, which is historically what they have done.
Yeah.
And I don't have to tell a soil scientist what bad news that is.
Yeah.
But our number one rotation in the Australian industry is soybeans. And so
Okay.
most of the farmers, my father included, they put the soybeans in with the intention of getting a cash crop off that. But in a situation like we've just had this year, we've had a very wet spring, a wet autumn or wet fall. And so a lot of those beans couldn't be harvested. So they get ploughed in, but then we do an offset on our fertilizer. And so we actually get some benefits out of that anyway.
Makes a ton of sense. So today on the Crop Science Podcast Show, we have Dr. Anthony Young. He's senior lecturer at crop protection at the University of Queensland in Australia. Now, Dr. Anthony, your focus of what you've already spoke about quite a bit is RSD, or the Ratoon Stunting disease. Let's go into that and its impact on Australian agriculture, and what are the steps to manage it?
Yeah, so this has been subject to a lot of debate, particularly over recent years. And just to make it really clear for your audience, it's been my side of the debate versus the industry side of the debate. So
Yeah.
when I did my PhD on this disease back in,
Hmm.
I started in the 1990s. When did I start? 1999. I finished in 2003. Which took me 4 years to finish. I found that the disease was highly underdiagnosed. So, just to describe the disease for your audience,
Yeah.
Ratoon stunting disease has no symptoms whatsoever. You can't look at the sugarcane and say, yes, this one has RSD, this one doesn't have RSD. And it took many years to work out that it's actually a bacterium that lives in the xylem vessels of the sugarcane. So your listeners would be aware that the xylem transports the water and mineral nutrients from the roots up through the
Hmm.
plant, right? So sugarcane that has Ratoon Stunting disease has an impediment into the function of that xylem. And so its impacts are worse when you have drier conditions. And that's how it was discovered. Back in
Hmm.
1945 in a place called Mackay in central Queensland, we had a heavy drought. At that stage, the sugar industry had released this brand new sugarcane variety, this Q28, and a farmer had planted half of his field with his own source of this Q28, but he ran out, right? So he asked the neighbour, he said, could I get some of those plants off you? The neighbour agreed, so he planted the other half of the field with the neighbour's Q28. And as I mentioned, it's a super dry summer. And when the farmer's looking at the crop, one was up here, one was down there, really quite a disparate situation. So he got a pathologist up to have a look at. They worked out, yes, it's the same thing, it's the same variety. And then they found they could transmit this disease. And so they thought for many years it was a virus because they couldn't isolate a bacterium. So RSD affects every single sugarcane variety, but some varieties are more resistant to others. And when I say resistant, they can still get the disease, but when they get the disease, they harbor less of the bacterium. And less of the
Hmm.
bacterium means fewer of the xylem vessels occluded, means less stunting, means less spread within a field. The disease is spread by planting infected cane and harvesting. And so if you can grow cane without planting or harvesting it, RSD will never be a problem for you.
Yeah.
And so my work revealed that the test that was being used for this disease, an ELISA test, really weak diagnostic platform.
Hmm.
And because it was really cumbersome to sample the cane, you had to cut the cane, blow out the xylem contents, and then do an antigen test. Farmers were only ever looking at their seed beds. They weren't looking at commercial crops. And so it was my thesis, as it were, that the disease was heavily underdiagnosed. And so,
Hmm.
without boring your listeners, after my PhD, I went and worked over in Ireland for a little while. Then I come back to Australia. I started working with the Department of Primary Industries. And when I left the DPI after 6 years, I got a job as a sugarcane extension officer. And during that time, I thought, well, I can revisit my PhD work here. And so I revisited a diagnostic platform and developed a new diagnostic that allowed me to test cane at any age. It was based on PCR. I could test many more stalks in the field. And when we start using this diagnostic, we see that RSD is vastly more widespread than previously. And so we had a situation where the sugar industry was claiming that RSD was present in fewer than 5% of fields.
Hmm.
Now we know it's closer to 40% of the fields in Australia. It's a big issue.
If you look across a field scale, is it a disease that's kind of mosaicked and pocketed within certain areas, or is it field wide?
Yeah, that's a really good question, Brian. We don't know. We don't know.
Okay.
We know that in a field, not every stalk will be infected, and not every we the sugarcane plant we call a stool. It's got multiple stalks coming out of it. Okay, we know that by its spread, we know that when a harvester goes through, it's going to spread it piecemeal, but not all the cane will be infected, and and that brings me to the closest thing to a field visual diagnostic we have.
Hmm.
The cane is up and down. If I'm driving at 100 kilometers an hour, let's say 60 miles an hour for your listeners, if I'm driving along and I'm looking at cane that's up and down and patchy, what's causing that? Could it be soil compaction? Could it be nematodes? Could it be poor weed control? Could it be rats in the cane? Could be all of these things. But given what I've discovered about the amount of RSD out there, it's likely it's not going to be RSD.
So I can assume one of the reasons the spread and the mass number of fields affected is for just heavily lack of identification, lack of knowledge that it's even there. Is that about right?
Exactly, and that's part of the battle that I've had with industry for many years. If you've got the foremost industry authorities saying, "Oh, the disease is present in fewer than 5% of fields,"
Hmm.
then the average farmer is going to assume, "Well, that's not going to be my fields. It's going to be someone
Hmm.
else's."
Yeah.
So I'm not going to do the right things. I'm not going to purchase clean seed. I'm not going to plant into fallow. I'm not going to do diagnostic testing because it's not worth my while. My while.
Hmm.
Um, That is slowly but surely changing, and we are seeing a changing of the guard. We're seeing senior pathologists who have been in the industry who have made these unsubstantiated claims. They've retired. We're getting a new breed of pathologists in. Our sugarcane breeders. This is where we need to fix RSD. We just need resistant varieties. But the argument against incorporating resistant varieties is that RSD is present in fewer than 5% of fields. We don't need resistant varieties.
Why spend money on breeding?
That's right.
Yeah.
And so what we then have is a situation where we're releasing these highly susceptible varieties, and in the breeding program, they don't have RSD because everything is hot water treated, which is a way of minimizing the disease. And then they land in the farmers' fields, and after 2 or 3 generations, they have high amounts of RSD. And so the yields start plummeting, and the growers are looking at that thinking, that's a crap variety. I don't like that variety. That variety is not as good as it, or something you often hear, that variety is not as good as it used to be. Nothing has changed in the variety. Something has changed in that pathoscape in which that variety is being grown. And so I guess my big issue with the current management of RSD is that it's having a lot of impacts that we haven't well gauged as an industry. And I'll just give you a couple of examples of those. When your listeners think of Queensland, they probably think the Great Barrier Reef, right? I think about the Great Barrier Reef.
Yeah.
The Great Barrier Reef is a global treasure. And I definitely recommend yourself, Brian, come over and have a look at it down the track. A lot of our sugarcane in Queensland is grown in catchment areas that run out into that Great Barrier Reef. Now, we've got a disease that's highly prevalent right throughout our sugar industries in these areas, right? And that means when the farmers get their second ratoon sort of thing, they think, oh, that cane's not very good. So then they hoe that cane out. As a soil scientist, you know that when you're hoeing, you're going to be releasing your organics, you're going to have eutrophication, all your nutrients are going to be running out, sediments are going to go into the waterways. What happens there is they form food for algae, which form food for things like the crown of thorn starfish larvae, which means more crowns of thorn starfish, which means more impact on those Great Barrier Reef catchment areas. A lot of your listeners are probably aware that the function of the reef, the barrier reef, you need high light penetration for it to work. To have that high light penetration, you have to have low nutrients. It's ironic. It's completely opposite to what you think. And yet if we're chucking more nutrients unnecessarily in because we are removing sugarcane that's infected with RSD, then we're really causing a bigger problem. And not just that, there's the farmer's sustainability, it's their profitability. To grow a ratoon crop of cane, a healthy ratoon crop of cane, doesn't cost much at all. You just harvest the cane, you might run a bit of fertilizer on, you might do 1 or 2 weed sprays. That's it, up it goes.
Yeah.
Versus the massive cost of removing a crop and then re-establishing a crop. So there's an economic, there's a social, and there's definitely an environmental element to better management for ratoon stunting disease.
So I guess a 2-parter on this one. If a farmer finds out that his crop is infected, can that crop be treated? And part C, what can be done? If it can't be treated, what are the steps to remove it from a farm?
Yeah, that's a really good question. It can't be treated. It's got to come out. Now, I mentioned earlier that historically a lot of farmers would hook out the cane and then they'd replant the cane. And we call that a replant crop.
Yep.
And this was one of the elements which led me to do my diagnostic work, because we know that there's a statistically significant higher amount of RSD in replant crops versus crops that are formed after a fallow.
Yeah.
And so I kept asking the industry, well, why is that? What does that tell us about, number one, the crop that was in there before, and number two, why it was removed? Okay, so we know that RSD can be transmitted. We don't know the mechanics of it. We don't know whether it's part of the rhizophagy cycle where the new sugarcane picks up the bacterium from the old stubble. We don't know whether it's existing in volunteers. When we say a volunteer, for your audience, when we take the cane out and let's say we don't completely destroy that crop, you might have a new sprout come up of the previous crop, right? That will be infected, of course.
Hmm.
And so we always, as an extension officer, I'd tell the farmers, if you have volunteers in your fallow, it's not a fallow. And
Yep.
so,
No.
yeah, and not just with Ratoon Stunting disease, but harboring nematodes, Pachymetra, other fusarium, other soil-borne pathogens. They've got their food source there. You're not breaking that cycle. Okay, so the only way to control RSD in the short term is fallow
Hmm.
and planting clean seed. And when we
Hmm.
say clean seed, this is seed that's certified to be clean. This is um, sugarcane is vegetatively propagated. We call it seed, but it's not seed. There's little chunks of sugarcane. We hot water treat that sequentially, year in, year out, so we know that it's going to be fairly as free from the bacterium as we possibly can make it,
Hmm.
and that's the only way to control RSD in the short term.
Is a single fallow season enough?
The economics of it, you're going to get more money for your sugarcane, and out of interest, you mentioned before we were speaking off beforehand, Brian, you're talking about Brazil. Out of interest, the sugar price in Australia and the world, I suppose, is highly linked to the oil prices. And so
Yeah. Oh, yeah.
when we have an increase in the oil prices, we have an increase in the sugar prices
Hmm.
because the biggest producer is Brazil. And Brazil, at the drop of a hat, can turn from sugar production to ethanol production.
Absolutely.
Okay, and that's an amazing thing, a part of their industry. Okay, so at the moment we have high oil prices, we have high sugar prices, so the farmers are going to be very keen to get the sugarcane back in. In terms of trying to break that cycle with just one fallow, one thing I was exploring a few years ago was the idea of doing a soybean fallow and then move into something like a sugar beet fallow. Now, we, as you're probably aware, there are tropical strains of sugar beet now that can be grown in Africa. So I was looking at, okay, we would have to re-engineer the front end of the sugar mill, but we could be producing more sugar and have that
Yeah.
third species to really break down, for example, the Pratolincus nematode association.
And you're not really hitting the sugar mill, which is a big societal issue because that sugar mill needs to be running to keep the people employed in the region. Or at least that's what I can assume from the mills that I've seen.
That is so spot on, and the sugar industry, for all its ills, right? And there are ills, okay.
Mm.
Um, when when there's money in cane, the communities do well. You see shops opening
Hmm.
versus shops shutting, and it's such a positive thing when you see a vibrant sugar community. And I'm looking forward, Brian, when you do come to Australia, introducing you to one of those.
Absolutely. Well, our time flew by already, Anthony. I'm going to be true. We're already out of time. I didn't expect it. This was a really fun time. So
It's time for our famous three. Did you know that on average you lose approximately 1 bushel an acre during harvest? Header loss and threshing loss due to wear and tear on your equipment can lessen your yield and profits. CNH Reman Wear Parts is a drop-in ready solution made from high-quality steel that lasts longer than the competition and is covered by an industry-leading 24-month warranty. CNH Reman is helping farmers have on-time delivery of their harvest. Visit your local Case IH or New Holland dealership for details.
that wraps up another episode of the Crop Science Podcast Show presented by Wisenetics. So, Dr. Young, a couple of our final questions before we really let you go here is, what's a go-to work resource? Where do you go when you have questions about what you're working with?
Oh, I'm going to be really honest. I'm a bit of a luddite. I dive onto Google Scholar. I
Yep.
like to see what other
Yep.
people are doing and how they've done it.
All right. Now, this is one I'm curious about. What do you do in your free time in Queensland?
Oh, I'm a father of three children, and
Ah,
all of them are avid bird watchers like their father.
yeah.
I love nothing better than getting into natural ecosystems and watching birds.
Oh, that's fantastic. And finally, where do people go to find out more about what you're doing at the University of Queensland?
Well, they can go to the University of Queensland site. Probably best off just email me. My email address is all readily available. I'm on LinkedIn and I'm on ResearchGate,
Mm.
but probably the UQ website is the best way to find me, Brian.
Well, appreciate it. Again, folks, this is Dr. Anthony Young, Senior Lecturer in Crop Protection at the University of Queensland in Australia. And all, if you enjoyed this video, share it with your friends, share it with your neighbors, get us out there, hit that like button, subscribe so you know when we have a new crop science podcast show released. Of course, leave any comments and suggestions you may have. Dr. Young, thank you so much for your time today.
It's been an absolute pleasure, Brian. Thank you.